Hi, well, welcome. I am Rachel Sanders, and this is my lab assistant, Allison. And today we are here brought to you by the Ohio soybean farmers and through Grow Next Gen. I'm one of their teacher leaders, so we're going to kind of do a little activity with you today to test the effectiveness of bio-based cleaners. Bio-based cleaners are now the new alternatives on the market to break down and get rid of that first layer of debris on your lab stations. So what we are going to do today is, as Allison is going to demonstrate, first we are going to need to label our auger plate, which is these nutrient auger plates labeled here in front of her. And as she is labeling, if you notice, she is actually creating a quadrant or a grid on top of her auger plate. And as any good lab technician would do, she is writing her initials. She is putting on today's date, because this is the day we are going to take our sample. She is going to be labeling with each trial in each quadrant, trial 1, trial 2, and trial 3. And then she is also going to have one quadrant left that's going to be a before as she swabs her lab surface before. So before we start here, we're kind of looking at how well does the bio-based cleaners actually break down that first layer or clean that first layer of grime off of your lab table. So with that, she is now going to take her ruler and she needs to make a little space that is 15 centimeters in length. So we're going to make a nice little box here for her to swab in the middle to test her cleaning skills and the effectiveness of the cleaner. And then we'll go 15 centimeters. And now here is her workspace that she'll be working through. Go through the edge of that tape. Now what she's going to do is she is going to take a sterile swab and she's going to make sure she opens it at the tip that has no cotton applicator so that it stays sterile and away from any bacteria or debris. And she's going to dip it into next the distilled water, beaker with distilled water. She's going to then place over her square that she has measured off to take a before sample of what kind of dirt and grime is there. And then next she's going to pick up her bacterial plate and she's going to actually swab lightly over the side that is marked the before side. And make sure, teachers, that your students do not jab into, that's a gelatin auger base. So you want to make sure they just kind of lightly go and scrub over top. Okay, so now we're going to clean your surface with your bio-based cleaner. Okay, and place you're going to extract 1 ml. And if you notice, she is going to go up to the first line. The tip to the first line is 1 milliliter of solution. And then she's going to pour over top of her lab surface and then grab a paper towel. And now you're going to wash your surface with your cleaner. Okay, you can place your paper towel to the side, get another paper towel, and you're going to need to dip in distilled water and rinse before we re-swab. Okay, and to wash your surface, you should rub around your cleaner really well. Now she is rinsing off with water and will lay her paper towel to the side. As it's air drying for a few seconds, roughly about 10, she will take a new sterile applicator and now she will actually rub across the surface in one area, and then that will be her first trial and she will swab her first trial on her plate. And if you notice, she is holding her lid at a 45-degree angle so that she tries to keep as much contamination as possible. Discard your applicator. She will take a new sterile applicator for her next trial, and this will be trial 2. And then she'll swab for her third one after trial 2. This gives you a good sample of the actual surface so that you can check to make sure your students are cleaning properly. Now you may ask, what is the difference between cleaning and sanitizing? Well, right now we're just worried about cleaning the surface. If you really wanted to also then go through a sanitation process, which is for an extension of this lab, then we would do another cleaning with an alcohol-based cleaner. But right now we're just trying to show how effective is the bio-based cleaner compared to your everyday soap and water. Okay? So now that she has swabbed all of her quadrants, she is now going to go and tape a Petri dish shut before we place it into the incubator. And she's just sealing, keeping away from any moisture. And she can either, if you're at school, you can store these in your incubator at 37 degrees Celsius, or you can leave them out on the counter for anywhere from 24 to 48 hours. And then we will look at them to see if there's any growth or if you cleaned up your surface correctly. Thanks again today for joining us on showing how to test effectiveness of bio-based cleaners. We would also like to deeply thank all the Ohio soybean farmers for their support in helping us create these activities. You can find these activities at grownextgen.org, and this lab is part of a unit called Soy Fresh, Soy Clean. So check it out and see what you think.